My 7th video in my AP Biology review series, and it is about cell communication. This entire picture shows communication between mating yeast cells. In the first part, mating factors are exchanged, which are chemical signals. Each cell releases factors which bind to the receptors of the other mating type cell. So in this case, we have A and alpha. Then, mating occurs, and a new cell is formed, so the nuclei of the two will fuse. The binding of a signal molecule, in this case a mating factor, causing a change in the cell in a series of steps is called a signal transduction pathway. Although yeast and animal cells have a very distinct common ancestor, their processes of signal transduction are similar, and this means that the process evolved a very long time ago. A cell will release a chemical signal, and these molecules are called ligands, which is a general term for a molecule that binds to another molecule, in this case a receptor. Ligands bind to a receptor on the target cell, and it's important to remember the specificity, which means that only specific target cells will recognize or respond to a specific chemical signal. Messenger molecules will relay the message, and then the cell will have a response. And this entire thing is called a signal transduction pathway, which is the conversion of a signal to a specific cell response. The first type of signaling we're going to talk about is direct contact, which includes cell junctions, where materials are transferred between two cells without crossing the plasma membrane, and an example of that is the top picture. There's also cell-to-cell recognition, where cells will communicate through membrane-bound molecules, like in the bottom one. Autocrine is where the cell signals itself, so it releases chemical signals that bind to the receptors on its own cell. Paracrine is when the cell signals a neighboring cell. Endocrine is when the cell signals over long distances. So in this case, hormones will be secreted, and hormones are chemical signals that are released and then will travel through fluid, which is usually the blood, in order to arrive at a target cell. Now for a little history. Earl Sutherland was a very important scientist who did a lot of groundbreaking research in this field. His research contributed to much of what we know about how chemical messengers relay information in signal transduction pathways. He discovered that the hormone epinephrine stimulates glycogen breakdown by activating an enzyme. Then he did many experiments and saw that glycogen was only broken down when epinephrine was placed into a solution with whole cells, and this told him several things. First, epinephrine does not communicate directly with the enzyme, and that intermediate steps must occur, and that the plasma membrane is involved in the transmission of the signal. His work suggested 3 steps: reception, transduction, and then a response. So this slide is going to be an overview of the 3. In reception, a signal molecule, which again is the ligand, is recognized and binds to a receptor. Then there's transduction. The binding of the ligand changes the shape of the receptor, and that's what starts transduction. It can be 1 step, or it can be several steps, where the signal may be amplified by a second messenger. Then there's the response, where the cell activity changes. That could be gene regulation, which could lead to a change in phenotype or production of a chemical. Now we're to go more in depth into each of these 3 steps, starting with reception. Again, a signal molecule will bind to a receptor. Most signal receptors are plasma membrane proteins. However, some of them are intracellular receptors, which means they're inside the cell. They could be in the cytoplasm or in the nucleus. And to bind to a receptor inside the cell, the signal molecules must pass through the membrane. Keep in mind that a large portion of the membrane is hydrophobic, that middle region. So these molecules must either be hydrophobic or very small so they can squeeze through the plasma membrane. Examples include steroid hormones, which, keep in mind, are lipids, so they are hydrophobic, and nitric oxide, which is a gas with very small molecules which can squeeze through the membrane. As the ligand binds, the receptor's shape changes. Now for G protein coupled receptors. Starting with that first part, when GDP is binded to the G protein is inactive. And for those of you who haven't heard of GDP before, you can think of it as similar to ADP, that's adenosine diphosphate. This is guanine diphosphate, so it's a different nitrogen base. As you can see in that top picture, when there's GDP, it's inactive. Then the hormone will bind, and that activates the receptor and causes a change in its shape, which allows the G protein to bind to it. GTP will displace GDP, and then the G protein is activated, like you can see there in the top. The activated G protein leaves the receptor and binds to an enzyme, which then activates the enzyme and can trigger a next step in the pathway. And that would be that last picture there. There are also receptor tyrosine kinases. Kinases are enzymes that catalyze the transfer of phosphate group. So in this case, a tyrosine kinase would be an enzyme that transfers a phosphate group to tyrosine, which is an amino acid from ATP. Starting with that top part of the picture, as monomers, the RTKs, which I just abbreviated, those are the receptor tyrosine kinases, they're inactive. When a signal molecule binds, a dimer forms, and a dimer is two identical molecules linked together. So as the signal molecules bind, they group together. The dimerization activates the kinase region of the protein to phosphorylate the tyrosines, and then the protein becomes fully activated. Specific relay proteins in the cell bind to the phosphorylated tyrosine, which becomes activated and then triggers a transduction pathway, which will lead to a cellular response. So as you can see, so far we've seen a lot of basically the domino effect, one thing affecting another, which affects another, and so on. And we'll continue seeing that pattern. There are ion channel receptors. In the beginning, the ion is unoccupied by the ligand and is closed, but the ligand binding causes the channel to open. Ions rapidly coming into the cell can trigger a change in cell activity. And when the ligand leaves, the channel will close again. The next step would be transduction, where signals are relayed to target molecules in the cell. Activated receptor activates another protein, which activates another protein. Again, that same pattern. Many times the signal is transduced by phosphorylation. Again, protein kinases we talked about earlier are enzymes that transfer a phosphate group from ATP to a protein. So as you can see in this picture, a protein kinase will activate another protein kinase, and that cycle continues. Protein phosphatases, which you can also see in this picture, are enzymes that rapidly remove the phosphate group from a protein. So when you see that dotted line and then the P, the phosphate group, that's caused by a protein phosphatase. Second messengers are small non-protein molecules or ions. The first messenger would be the ligand that starts everything. An example is cyclic AMP or CAMP. AMP is adenosine monophosphate, which is made from removing 2 phosphate groups from ATP, adenosine triphosphate. The reaction is catalyzed by adenyl cyclase. Cyclic AMP turns into non-active AMP by the addition of phosphodiesterase, another enzyme. CAMP will often activate protein kinase A, which will in turn phosphorylate another protein. As you can see in the picture, we have a G protein coupled reaction where the ligand binding will cause the activation of the G protein, which will then activate the enzyme adenyl cyclase, in this case adenylate cyclase, they're the same thing, which catalyze the reaction, the creation of CAMP, which will activate protein kinase A, which will then phosphorylate another protein. As you can see, again, these pathways are similar to a domino effect, where one thing causes the next to be activating, and this is all relaying the initial signal. More second messengers include calcium ions, which are commonly used as a second messenger. In animal cells, they're important. They're important in plant cells as well. They cause muscle contraction, secretion of certain substances, and cell division. In plants, it can cause greening when in light. The concentration of calcium ions is low in the cytosol, but high in the endoplasmic reticulum. So adding a bit into the cytoplasm would greatly increase the percentage of it and can cause the cell's activity to change. Some signal transduction pathways involve pumping calcium ions out of the ER and into the cytosol, and this will be caused by other second messengers. If you look at this picture, there's a bunch of complicated abbreviations and things that we don't really need to worry about. I think the important part is to realize, again, we have the ligand binding, and then a bunch of second messengers activating other second messengers, and then the final response is calcium ions being released from the endoplasmic reticulum into the cytoplasm, and then that will result in a change in the cell's activity. Calcium ions will still be called second messengers because scientists still use the term second messengers when referring to all small non-protein parts of a signal transduction pathway. So even though it's not really the second messenger, it could be like the third or something, they're still called second messengers. Then there's a response where the cell will have a change in activity in the cytoplasm or regulate transcription. For example, liver cells will break down glycogen because of the hormone epinephrine. The final activated molecule could be a transcription factor, which could be regulating the production of an enzyme or another molecule by turning genes on and off in the nucleus. In this picture, CREB is a transcription factor, which is binding to target genes in order to activate them to create the needed protein. There are 2 types of feedback, negative and positive. We're going to start with negative, which maintains homeostasis. It helps return systems to normal if conditions are disturbed. An example would be blood sugar regulation. Insulin is released when blood sugar levels are high, and glucagon is released when blood sugar levels are low. And you can see that pathway there at the top. There's positive feedback, which causes a change to be amplified. So as you can tell, positive feedback is the opposite. And the system would move further away from normal. For example, blood clotting, where activated platelets activate other platelets. And again, all of feedback is controlled by signal molecules, so they all do tie in together nicely. That's it for this video. Thank you so much for watching, and please subscribe if you would like to see more videos.